So I'm proud and happy and pleased to announce Professor Cowan, who is my previous PhD advisor, postdoc advisor, my current collaborator, and a very close friend, and one of the best, smartest, and best human beings that I have ever known. Okay, so Noah finished his Bachelor of Science degree in Electrical Engineering department in, I don't know, many years ago. And then he moved on to the Michigan University and completed his master's and PhD degrees again in electrical engineering. And then he moved to Hopkins to become a professor in mechanical engineering department. Me and Noah published a paper about the symmetry in human walking. We kind of like symmetry. In order to keep symmetry, since I'm a mechanical engineer, I came to the electrical engineering department such that we maintain the symmetry in terms of the path. I did master's in electrical engineering, so it's not symmetric perfectly, but it's symmetric enough. Okay. Now currently he's a professor in Johns Hopkins University mechanics department with appointees in electrical engineering, biomedical engineering, and neuroscience. And he is also the chair of the Hopkins Robotics Center, which is called Laboratory of Computational Science and Robotics. And I'm also deputy chair here in Robotics Center. Again, we are still keeping the symmetry as far as possible. I'm currently an assistant associate professor. We have things to do. Okay, Noah earned many of the awards. The good thing is he earned many of them after I went to the Hopkins. So I have a part in that. So he received the NSF case, I forgot, case, right?
Yep.
Award. And he is one of the most unique awardees in this theory. Normally it's given even number of, I don't know the number, but in 1 year it is given to the odd number of the awardees, right? And you are the single unique one. It has a nice story, or it depends. And also Noah received a Discovery Award at Hopkins, which is, I think, very prestigious. William Hughes Award for Excellence. And I think, yes, McDonald Foundation Scholar Award in Complex Systems, which is nice because I was funded from this award as far as I remember. And one of the best awards is kind of the Down Payment Award, conferring having an external positive impact upon the lives of one or more undergraduate students. Actually, this is true for the award, but I think this is true for any people work with Noah Cowan. Okay, student, undergrad, whatever else. I was really spent very good 6 years at Baltimore, which is kind of a very good city, but it has problems. But 6 years as a graduate student is hard. But Noah made our life is very nice, both from the technical point of view and both from the human point of view. So, and I told you, many of you, that this will be one of the best presentations that you will ever see. And it is true so far, and you will see that. Okay.
Well, thank you, Mary. Thank you for that incredibly flattering introduction. And it's really been a great privilege to come to Ankara. I have so many friends and colleagues and collaborators here, former trainees. It's really a kind of walk through, really a kind of history of my career, starting from my graduate days, where my lab mate was Ulrich Saranla. Actually, even before my graduate days, one of my first teachers of control theory was Hitai Osbe at Bilkent, who's now at Bilkent. He was a professor at Ohio State when I was an undergrad, and I took my first control theory course with him. And so he's who invited me to Bilkent earlier this week. And then I also have had an extraordinary opportunity to have in my lab trainees. So I have really sort of the past, present, and future, if you will, of my life represented in this one incredible and intellectually vibrant city. So it's been great to come back. And in terms of having impact on my students' careers, and I'm very grateful that my students feel that way, the extraordinary impact that, make sure I'm on, the extraordinary impact that the students have had on my career, can't be understated. This is just the publications from these two trainees that I've had in my lab, which is an amazing number of papers to come out of just two people. But it also includes collaborations with Ulich and Omer, and Hitai is on here somewhere, and many collaborators in Turkey. And so it's just been an incredible opportunity to come back and pay a visit, and maybe some way return the favor of the incredible impact that you guys have had on my career. So just a little bit of a tour of the work that I do in my lab more broadly. So I got my training in electrical engineering at Ohio State and then Michigan. but I've always been interested in how things move since way before I went to college. In middle school and high school, I became a juggler, actually competed nationally in the United States as a juggler, because I was just fascinated with how things move. And I sort of took a very practical approach to learning how to do that. And so maybe that juggling is what gave me an intuition for the way organisms interact in the world. I don't know. But when I went off to graduate school, there was a professor there by the name of Dan Kodacek, who was also Ulrich Saranda's advisor, who had one of the world's best, actually the world's best by far, juggling robot at the time. And I thought, oh my gosh, you can use control theory, which I had learned from Hittai, among other people at Ohio State, and mix it together with my life passion of juggling, and really understand the dynamic interaction of feedback systems and their environment. But during that time, I began to notice the incredible world of biology, where evolution has spent eons really perfecting the art of dynamic interaction with the environment. And so the first lowly organism that I studied was when I was a postdoc in Bob Full's lab, studying this cockroach, Paraplaneta americana, running along a wall as fast as it could, using just feedback from its antenna to control its movement. And so we modeled that all the way down from the roles of individual microscopic hairs on the antenna up to the whole body locomotion mechanics, neural recordings, and so forth. Project that Merritt is quite familiar with is the human motor control work that I do with Professor Amy Bastian at Johns Hopkins, who actually was just this week elected to the National Academy of Science. I don't know if you heard that, Merritt, but she's an incredible scientist. And this is the project that Merritt was talking about, where we have a paper called Human Walking is Symmetric, and then in parentheses, enough, where we modeled the locomotor dynamics of human walking. And then one of the organisms that I'm going to tell you the most about today is the weakly electric glass knifefish, Eigenmania virescens. I don't see the green border around the screen. I want to make sure you guys can see that still online. Okay, great. Okay, great. There was a green border that's kind of disappeared, so I wanted to make sure. This is a bottom view, and we're going to see a lot of this fish today, but fish swimming forward and backward. You might have seen Ismail's talks on this amazing organism, swimming back and forth to track a refuge, among many other organisms. Here's a project, actually, that as I was looking over the history, Merritt was at some point involved in this as well, looking at the abdominal visuomotor response in hawk moths. This is a hawk moth being given a visual stimulus. The moth is glued to the end of a stick, and as it's given that visual stimulus, it moves its abdomen, which is a very large inertial part of its body, in response to the visual stimulus. And what we discovered is that the abdomen is used as kind of a self-balancing mechanism. Imagining that you have a quadcopter that has its battery down below, the reaction torque caused by moving the battery reorients the propeller system, if you will. So we actually did a little robotic demonstration of that while Merritt was in the lab. Because I was trained as a control theorist, the way I approach these biological organisms is through the lens of control theory. And so with control theory, we like to describe things in terms of little boxes and arrows that come in and out, where we actually mean something very precise, which is that there's some signal that is sent into a dynamical system. That dynamical system evolves according to some, say, system of differential equations. And out of that comes another signal that propagates to another box, and so on. I want to come back to the fact that these arrows are one directional in this diagram. That's a big assumption we make in control theory. But as a feedback theorist, we recognize that perhaps the most important cause of sensory stimulus to any robot or animal is the consequence of its own movement. So as I turn my head, I see a massive retinal shift of the visual world on my retina, a rapid shift of the visual world on my retina. As I move, the vestibular system in my inner ear, my inertial navigation system is more heavily excited by my own movement than by something in the outside world. And so what we do to try to understand this from a biological context is we do system identification. We move some sensory scene in the environment. We change the sensory conditions for the animal, something like adding noise or taking away noise to the sensory environment. We provide mechanical disturbances, pushing on a system, if you will. We change the locomotor dynamics, change the substrate that an animal was locomoting on, change the flow conditions, and so forth. And then we also do things like open and close feedback loops, because we're control theorists and we can do really amazing things like Ismail's doing, where we're measuring an animal in real time and we can feed it back through our apparatus to either open or close feedback loops. And then through that process, we're also taking electrophysiological recordings inside the nervous system of the animal, measuring the muscle commands, these are so-called EMGs or electromyograms, measuring the forces with force plates if it's a human locomotion running on a force plate, or a cockroach running across a force plate. We can measure the forces. We're doing that now with our rat project. We measure the kinematics using videography, which has gotten a lot easier thanks to deep learning based methods. But I mentioned that when we do this, we take this control theoretic approach, which implicitly assumes, because of the way we were trained for 100 years, that dynamical systems are one-dimensional processing units that take information in and send information out. But anybody who's taken a circuit theory class knows that one of the first things you learn is how to create a Thevenin equivalent model of a circuit that has an output resistance. And if the input impedance is not high enough, then the voltage that comes out of your first circuit will change as a function of how much current is drawn by the second circuit. So you really need to have amplifiers between these systems in order to allow a one-directional flow of information. And so I've been bothered by that for actually a really long time. And I remember in 2010, I met Eduardo Sontag at a control theory and biology workshop. And he was explaining to me, it was a bunch of biologists and a few control theorists. And we were all interacting and trying to understand how control theory could teach biologists new things and how biologists could give control theorists new ideas and new theories. And he had developed a theory with some really well-known cellular biologists on how creating those one-directional arrows at the cellular level, where in order to signal, you have to create some protein. That protein then gets consumed by the downstream process, changing the signal that you're actually trying to play into the system. Creating amplifiers can be done at the cellular level, but it turns out to be very metabolically costly. And so I thought that's really, it sort of planted a seed in my head. And years later, as I was drawing block diagrams for one of our biological control systems, it occurred to me that that one of the reasons we get away with these one-directional arrows is that muscles are actually really amazing amplifiers. The electrical signal that comes from your nervous system is not affected by the forces and motions that play out through the muscle. So if I measure an electromyogram, I know the input to the mechanical system, and that is not affected, that's not loaded by the mechanics. And on the other side, many sensory systems, although this may not be true for touch, but many sensory systems like vision or electrosense, like I'll talk about in a little bit, are also these really nice high gain amplification systems where my motion does not affect the motion of the external world. And so I get to measure my relative position, and I get kind of a one-way flow of information that way. My motion doesn't directly affect the outside motion except through my control effectors. And so this makes neuroscience an excellent sort of two-way street with control theory, which is that not only can our models actually apply very well to trying to understand biology, but it makes biology a better teacher for new ideas in control theory, because it's an incredible mathematical convenience to separate out the dynamics in this way. Along those lines, as a control theorist, we often think about, going forward, maybe I have to click, or just slow. We often think about the musculoskeletal plant. That's the control theory jargon for the system that's being controlled. Although since I'm going to be talking about animals, often the plant is the animal, which is kind of a pun. The musculoskeletal system is really the driver in many ways of my thinking about control theory and biology for a long time. And we spent a lot of effort, and actually Ismail did some really pioneering work in my lab on understanding the musculoskeletal plant in our fish that we studied. And then with that model, we can then have better hypotheses, optimal control, robust control, things like that, PID control. Like what's our model for how the nervous system might be doing some particular control action is much better informed if we have a good model of the plant. And so the animal that we study is the weakly electric glass knifefish Eigenmannia virescens. I really feel this is nature's gift to a control engineer. It's really an extraordinary animal that gives incredible affordance over our ability to manipulate and measure all throughout its physiology to try to understand how it controls behavior. But also as a control engineer, its name is eigenmania. And, like, I'm just trying to figure out its eigenvalues for my entire life. So let me just show a little schematic. I was lucky to have a student, Itai Roth, in my lab, who was a fine artist before he became an engineer. And so I have a lot of really great pictures of animals because of him. Also a lot of great research that I'm going to tell you about really originated with him. So here's the glass knifefish. It has a number of adaptations that I mentioned are very good for control engineering. One is that it has image forming eyes, just like very similar, analogous to our eyes. They're able to map the three-dimensional world onto a two-dimensional sensorium. But in addition to that, they have a separate, physically independent way of measuring the three-dimensional environment, which is a system of electroreceptors over the surface of their skin that measure the voltage difference between the outside of the fish and the inside of the fish over its body surface. And each one of these stipples is supposed to represent an electroreceptor, basically a little voltmeter. And in the tail of the fish is an electric organ that's an evolutionary adaptation of a muscle that pumps ions and creates an oscillating current source, low voltage, but similar to an electric eel that could shock you. This generates an ongoing electric field that envelops the animal, and then objects in its surrounding distort that electric field, change the voltage on the skin, and allow the animal to feel at a distance the objects near it using this electrosense. So 2 independent sensor modalities is very rich for perturbations and manipulations. And then in addition, if that's not enough, they have this elongated ribbon fin on their ventral side. It's actually something that I'll tell you a little bit more about, but I want to mention one thing before I go on, is that a lot of engineers enter the world of bio-inspired engineering, assuming that because evolution, we design evolutionary algorithms to optimize things in engineering, that evolution has been going on so long that these animals must be optimal. That could not be further from the truth. Evolution works on the just good enough principle. Things need to survive in order to survive. It's kind of a tautology, but also the best you could say is that it's a optimization-like process working against a changing cost landscape of where the cost function is survival. And not only is that cost landscape changing, but it's working based on an evolutionary history, a baggage that it has to carry forward from generation to generation. Whales have pelvises, but I'm not sure pelvises are particularly optimal for swimming, but they evolved from tetrapods. Tetrapods were land animals. They evolved from those, so they have a leftover part that is apparently not affecting them too badly, but it doesn't necessarily mean it's a good part of the design. So if I wanted to look for locomotion efficiency, I might not think pelvises. That's what I need to add. Animals have them and they're optimal. This is a ribbon fin. It's an anal fin, which was evolved from a fin back here. An anal fin is situated behind the anus on a fish. This thing evolved by tweaking that anal fin to become long. And so now this fish poops out of its neck. In no world is that optimal, but it evolved from tweaking a previous design, right? And it was not able to change the topology of the digestive system to move that cord somewhere else, I guess, through the evolutionary process. So it's still very good as an overall design, but like our designs in engineering, it often has weaknesses based on prior versions that we developed. So, but we as engineers can wipe the slate clean. We can see good aspects of a design, recognize those as that evolution does find creative solutions, and then we can be engineers and optimize for our purposes and take it even further than the animal is able to do in that regard. So I mentioned that they generate this ongoing weak electric field somewhere around 400 hertz and very low amplitude, a few millivolts. And this is just a nice illustration. I think this is out of Heiligenberg's book, but I don't remember. This definitely is out of Heiligenberg's book. He was one of the pioneers in studying electric fish, and this is showing a top-down view illustrating the current lines going from an electric dipole that's inside and outside the fish in this big organ in their tail. And if there's a nearby insulating object, it distorts those current lines, distorts the electric field isopotentials, changes the voltage on the skin, and the animal says, aha, there's an obstacle near me. The ribbon fin, I hope you can see in the back, is this beautiful structure. This is a bottom view. You hope you can see these two inward waves that are propagating and meeting at the middle. all day long, every day, these fish sit in tanks generating these counter-propagating waves resisting each other, which might seem counterintuitive, but I'll come back to that in a minute. Seems potentially energetically wasteful. But perhaps the most convenient feature that these organisms have from a control engineering point of view and trying to understand how they control and regulate behavior is that they like to hide in little tubes. So this is my colleague, Eric Fortune, standing up 2 floors up with his hand holding onto a plastic tube, and he's going to be moving it back and forth now. And that fish is not captured. It's not connected to that tube. It can swim out at any time. But it looks to me, every time I watch this video, and I've seen this video for many, many years, that fish is attached to the tube by a spring and a damper, like a PD controller, actually. That's actually nothing but the mapping between sensory inputs, some internal computation of the brain, and motor outputs that gives the appearance of being a spring damper system. And of course, it's not, Eric would get very tired if he had to do all of our experiments. So instead we use a electric motor to move a refuge back and forth. We're now looking at a bottom view of a similar piece of PVC pipe that's been cut off, positioned at the bottom of a tank. We're videoing it from below, and we can see the little pectoral fins here and this ribbon fin moving very quickly. They're about 10 hertz, 10 cycles a second for any one up and down wave of any one part of the fin. And we're tracking over time the position of the refuge and the position of the fish. And you see they track very well. And from that, we can do things like get a gain and a phase and do a frequency response plot. And since they're operating near an equilibrium, nonlinear systems are, everybody says, well, how are you using linear systems theory? Animals, biology is incredibly nonlinear. Yes, that's true, but the same reason we can use linear systems theory for many problems in control engineering is that systems operating near equilibria, by and large, should be locally linearizable to some domain. Turns out there's very interesting ways in which these animals are nonlinear, even locally, which means they have non-hyperbolic equilibria, which is kind of interesting. And maybe that's actually useful because perhaps having eigenvalues near 0 and using nonlinear controllers gives you kind of an ability to do to escape kind of stability, maneuverability kinds of trade-offs, if you will. I'm not going to talk about that today, but that's an interesting point of discussion. But we can still actually get very good linear models as long as we use noisy inputs. They're very good at predicting signals, just like we are. If you give somebody a very regular signal, you can sort of lock onto that. You develop a model for that motion, and that model allows you to really reduce your phase lag and get your gain correct in a way that reveals a nonlinear computation, a nonlinear adaptive internal model principle kind of controller that is the subject of my current student's PhD dissertation. So this is a video that I'm going to show that was analyzed by Ismail Uyunik in a very nice 2020 eLife paper, and I'm going to orient you to it. A little bit hard to see, but you're going to see lots of repetitions of it. So this is our fish. We're going to zoom in on this black box, which is shown here. And the Ismail processed the video so that the fish would remain stationary on the video screen, even though it's swimming back and forth. And so we're going to represent its velocity by the length of this arrow. And then I mentioned that the fish generates these two sort of like inward counterpropagating waves that meet in the middle. The fish is doing that, it's not like a rope where you shake one end and the wave propagates. It actually has 140 fin rays down the length of the fin that are individually actuated by sort of push-pull muscles and antagonistic muscle pairs that move each one of these up and down. And then the nervous system sends those commands in a way that's phase related to each other so that they generate a metachronal traveling wave, okay? But what's cool, that's as if that's not cool. What's really cool is that they can change how much of the fin is going one way versus the other way, so that more of the fin can be propagating its wave this way and less of it the other way, so that it creates a differential force and causes the fish to swim one way or the other. That trimming of that middle nodal, we call it the nodal point back and forth, is the source of control. So you're going to see his digitization of that nodal point moving back and forth here. And it's going to first show several repetitions of it going in full speed. It's very hard to see what's going on, so he's going to slow that down for us. And he's actually going to show the peaks and troughs of the traveling wave coming in together and meeting at that nodal position. And now we can actually imagine finding a dynamical system, a plant model that relates the movement of this nodal position to the movement of the fish, sort of a system ID. And here it is at 10 times slowed down speed. And this was before we were really using deep lab cut and these other automated deep learning based machine learning. So an undergraduate clicked 50,000 times on our video data in order to capture this data. Those days are fortunately mostly behind us. But we can see now if we take a snapshot of the fish and show it here, and here's this nodal, and we trace these peaks and troughs over time, that's these sort of diagonally inward lines. We can then look at the nodal position over time as the fish is swimming forward and backward, and then do many, many replicates of that. And then take the average nodal position trajectory and the average fish trajectory in blue and estimate a frequency response for that frequency. And it turns out that was Ismail's work, and it turns out that compared very well to a first principles model and biological finding that we found for a static situation. It's a little hard to explain, but we published in PNAS. Ismail was able to verify that dynamically using system ID. We have a first principles model that was tested with a robot and with numerical simulations that he then validated using system identification in this really lovely work. So we've been working on this animal a long time. Our first paper was in 2007, but I actually started working on electric fish in 2004 when Eric Fortune and I collected our first data. It just took us a long time to get our head around it. But since then, we've really published a wide variety of findings on the plant, the controller, active sensing, social dynamics, like a rich array of analysis of this fish's behavior using system identification and control theory. You see Ismail's name in there as well. He's made several contributions to this. So we just zoom in here on the plant and controller diagram, a highly simplified and stylized diagram that still provides a lot of insight. And look at just a few of the people that have worked with us on this. This is not an exhaustive list, but some of the most impactful work was done by these folks studying the plant and the controller. We can actually summarize surprisingly simply the locomotor dynamics using Newton's laws together with a damping term that comes from these counter-propagating waves and the difference of quadratic forces. You can check that out in our paper. I know that Ismail's work is now extending that. And then the nodal position provides this control input, which is the next best thing to an electromyogram because it's this, the bandwidth of that ribbon fin is 10 times higher than the bandwidth of the organism, allowing us to use its kinematics as a virtual input to the plant. So even though electromyogram recordings are hard in these fish, although we have tried, we can use these as a surrogate for that. Okay, so I mentioned that we drive our analysis based on the plant, but what about the sensors in these organisms? They turn out to play an incredibly important role in shaping behavior and might challenge some of our notions in control theory. So properties of sensory receptors, such as not just the sensory receptors, but sensory systems like occlusions, for example, require robots or animals to make physical movements in order to sense their environment. If I want to see where I put my laser pointer, for example, I have to look behind my monitor and see if I set it back there. I have to actually do motor actions in order to sense information. I can't just take information in passively. But there's more subtle and nuanced characteristics of receptors that cause animals to need to do that. So for example, our sensory receptors are very good at measuring change, but they're not very good at absolute. So they're precise, but not accurate, right? And in fact, they tend to block sensory information. So I'm sure all had the experience when you're working in an environment where there's an HVAC system running and it's loud, but you've just forgotten about it. And then it turns off and you're like, whoa. So like it's almost deafeningly quiet because your nervous system had kind of learned to ignore that background noise. It's the same reason you don't feel your clothes as you're sitting in your chair because your skin receptors adapt to that persistent stimulus. And so in order to sense something like texture, we don't just put our hands on something. We take something in our hand and we rub our hand back and forth and we feel what that feels like. Or if we want to measure the weight, we take advantage of the deeper receptors in our muscles and our skin and so forth, and we move the object up and down to do active sensing to give us more information about the weight, or to measure temperature, we'll press our receptors onto it so that we can transfer heat from one source, from one object to our hand, or vice versa, depending on which is cooler, to get change in our receptors, right? And so how do we do that? We have a number of different strategies. My former postdoc and Eric's former PhD student, Sarah Stamper, a little cut off on the left, but I think you'll be able to see everything. Wrote a nice review paper describing many of the different strategies organisms use to seek information. But it's basically strategies that organisms use in which self-generated energy is used to sample the environment, and it can be done, used, for example, as I mentioned, to do things like overcome sensory occlusions. And why we do it is, one is that information in the environment is distributed. So I mentioned sort of the occlusion problem, for example, but more subtle is this adapting high pass nature of the sensors. And so exploratory movements can be used to enhance sensation, and that's a theme that's going to come back throughout this talk. And the challenge is, and this is where control engineers, I think, don't have a good handle, is how do you take the same motor apparatus and use it to simultaneously solve the problem of getting information, which is exploring, and using information for a task, which is exploiting, even when those particular problems of explore and exploit might be at conflict with each other, because you're using the same system. How many people have had a linear systems theory course? Okay, good.
Yeah,
do you remember? Good. Okay, this is a test then. How many people know what the separation principle is? I got one hand. Come on. How about pole placement of observers and controllers? Okay. Do I care what the observer poles are when I place the controller poles, or vice versa? You can place them independently, right? There's this separation principle. I don't worry about sensing. I just design a state feedback controller, and then I design an observer, and I do those two things independently, and I can even make that optimal with a linear quadratic Gaussian controller under certain assumptions. And I just use a common filter and a linear quadratic regulator, and I don't worry about either one as I'm designing the other, right? And then you do some things, if you're really clever, you do some loop gain recovery and some other things to make the system look more like a linear quadratic regulator. So it's not the whole story, but most of the story is the separation principle. That is not the case in biology. Animals are constantly using motor signals in order to sense, to get information, in order to do stuff all the time. Like, it is the rule, not the exception. So now let me give you a really salient example. Might be hard for the folks in the back, but try your best to focus your eye on this plus sign and try to keep your head and eyes as still as possible. Do not actively sense. Just look at that plus sign. And then look around and see that the purple didn't really disappear. And if you experience the illusion, if you weren't able to, then just go look up Troxler fading, kind of a small URL there, but look up Troxler fading, and you'll see this example, and you can try it. Your sensory system begins to adapt out that peripheral movement until you move your fovea around to identify it. And there's something special about the way this little thing is moving around that I don't understand the physiology for, but it illustrates the point of the need to actively sense in order to refresh your sensorium. So now let's look at how the fish do that. Let me just check my time. Let me look at how the fish do that. This is the bottom view of a fish. In infrared illumination, the lights are on. You can't tell the difference between lights on and lights off because it's infrared, what we're recording in. And here's the fish moving back and forth. That's the fish, and this is the refuge. At some point, the lights are going to turn off. And we'll see a categorical change in the fish's 4-act swimming behavior. And this is what the graph in time looks like. The refuge is the red trajectory, and the fish is the blue trajectory. And with some exceptions, it's mostly just tracking. And then as soon as the lights go off, it's still tracking, but it's got this ancillary movement, which is active sensing. And we've got a lot of great evidence for that across a number of papers, that that's really an active sensing behavior, and not just it's the lights are off, let's party. It really is active sensing. And here's the opposite. We're going to start with the lights on. It's doing this oscillatory movement. Occasionally not. Sometimes it just floats along with the refuge. That's really important observation. And then boom, the lights go on, and it tracks like really smoothly. Okay, so Sarah Stamper did some beautiful work on that. That's her paper. So how can we model and understand this? We're control engineers. So let's take simplified sensory scene, S of X. In this case, it's just a sine wave grading. So S of X might be the 0, 1, 0, 1, a sine wave of some sort. And then let's just think about one receptor on the surface of the skin. And we're going to say S of X1. X1 is going to be the first state variable of our system, which is the position of the fish. And so we're going to have one sensor that, as a function of the fish's position, measures the scene. So it's nonlinear already because I now have the output being a sinusoidal function of the fish's position. Okay. But locally, it could be linear, like say near an edge, it could be a linear function. S of X could just be, say, X. Now, let's take this simplified plant model that Ismail made substantial contributions to, and there's actually a typo here, there should be another X. X dot equals AX plus BU, just a simple second order mass damper system, which is this validated plant model. And then the sensor output, we're going to take as a high pass filtered version of the sensory scene. The simplest high pass filter is a time derivative. So that's what we do. Just to illustrate the idea, the time derivative of the sensory measurement as a function of the fish's position gives us this particular relationship. Now, it turns out if you linearize those dynamics, which is something that after taking Merritt's class, I'm sure you can do with your eyes closed, you've already calculated that this is an unobservable system. So it's unobservable as a linear system near an equilibrium point. No matter where I can position that sensor right in an edge and try to stabilize that, and I cannot design a linear controller that can do that. Okay, so the question is, could I design a nonlinear controller that could do that? Could I take, different state variables, X is position, C is velocity, so that's like my X1 and X2 in the previous slide. Could I take this nonlinear dynamical system, which is the same nonlinear dynamical system, take a measurement, and I allow myself any dynamical feedback, linear or no, doesn't just have to be a state estimator or state observer, any dynamical feedback, and create a closed loop system. Can I stabilize an equilibrium point? And the answer, in the surprising one-line proof, is that no, you can't stabilize the system. That's in our archive paper with Eduardo Sontag. And so you think, well, it's hopeless. I can't stabilize the system around equilibrium. Well, the fish doesn't stabilize either. It oscillates all the time. But maybe I can do well enough. Maybe with this high pass sensor, I just need to add a little bit of extra motion, and then I can estimate my environment, estimate my position relative to the environment, and control my location. And so the part where Eduardo contributed quite a bit was to show that this system is actually globally observable as long as that scene is not periodic. So the example I gave was periodic, so that's not globally observable, but that just means that I wouldn't be able to tell the difference between which part of the sine wave I was on. I would only know my position up to the period, right, for a periodic signal. But if I have an aperiodic signal, which in general, over long enough distances, the environment is aperiodic, then I can identify my location, okay? That's a technical, but nice technical contribution that Eduardo made for us. So now how do we disentangle the problem of controlling my position within the refuge and moving so that I can sense the refuge and my position? I have to move to sense where I am in the refuge, and then I want to control my position within the refuge. How do I do that? So so that's the task level controller that controls where I am, and the active sensing generator seeks to capture information. And so we have a lot of work looking at the task level control, a lot of work looking at the active sensing, including also some more work by Ismail. I tell you, these guys just make this like widespread contributions. Please keep, the great students and trainees coming. It's been so much fun. So my current student, Devjyoti Biswas, is taking this to the next level by really trying to understand this trade-off between explore and exploit. And so I'll just tell you briefly about that. I've given you the setup. So he's going to look at a case with and without vision. Electrosense will always be present, but he'll compare vision and no vision. And the data here was collected by my other graduate student, Yu Yang, and analyzed by Devjyoti. And so we're going to talk about exploit as being somehow tracking the refuge, and explore as moving relative to the refuge. You've already seen this video. And so the important thing is that the explore and exploit both use the same ribbon fin locomotor system. They're both in the same linear dimension. and they conflict with each other, as I mentioned before. So how does the animal resolve that? So we're going to simplify things by, instead of tracking a moving refuge, we're going to keep the refuge stationary, in which case exploitative movements just become the small corrective movements needed to stabilize position. Subject to little perturbations in the water, we've got our little air bubbler running in the corner. The motor system has errors, it's not always perfect, so that'll perturb the animal. It's called motor noise, the process model noise, sensor noise, and so on. And then the exploratory movements will be these oscillatory ancillary movements that you see in those videos. And there's evidence that dates back at least to the 18th century that these really are truly for the purpose of sensing, all the way up to our own current work. And our hypothesis was that the animal would continuously inject a persistently exciting signal. If you're familiar with the idea of persistence of excitation, a persistently exciting signal that would allow it to do state estimation for control. And so here's an example trace in the light where the fish is doing sort of exploitative movements for the most part, and it does these large sort of occasional movement bouts within the refuge. And we're going to focus on movements in the X directions. So X being, in this case, I've drawn it up and down. And then in the dark, we see quite a different path that the animal takes as it's doing the station keeping task. These very large exploratory movements, but interdigitated by these flat segments, and that's going to become really important. So here's the trace in the light, and it's time derivative, and Yu Yang spent a tremendous amount of effort really making sure that these data were much more accurate than if you use an off-the-shelf like deep learning algorithm. It's really good classical style sub-pixel accuracy tracker at 100 frames a second, and really careful, great care is taken in computing the velocities, because all of our analysis is going to be in the velocity domain. So we spent better part of 6 months really making sure this instrumentation was accurate, because we were seeing results that frankly I didn't believe. So I made him really make sure the instrumentation wasn't lying to us, because it just violated my intuition. And you'll see why in a second. So what we say is that the exploitative movements are these small, like auto-regressive moving average kind of things, where the fish is getting perturbed by sensory motor noise and recovering back to its goal. And then occasional large exploratory movements. And in the dark, we see the same thing, but the exploitative movements are less frequent, or I should say the exploratory movements are more frequent. So our persistent excitation idea seems to be wrong. Like, it's not just constantly injecting noise, it's injecting noise occasionally. And so our second hypothesis, after kind of ruling that out, was that maybe the animals are actually turning on and off these exploratory movements. And so the way we look at that is we take this is now 7 trajectories in the dark and 10 trajectories in the light from one fish of the velocity overlaid on top of each other. And we can compute a histogram of those by projecting those velocities to histogram bins that we're putting on a log scale. That may not be easy to see for you, I'm sorry. It's a log scale. And for reference, like a Gaussian distribution on a log scale, log of e to the minus x squared is a quadratic. So we expect to see, if this was Gaussian, a quadratic curve on this histogram, okay? But we don't see that at all. We see this distinctive sharp peak with these broad shoulders. And so we're going to refer to this as a broad shoulder distribution. It's kind of a colloquial term. It's not a precise term in the literature. It's not a heavy tail distribution, which has something to do with the ratio of the fourth and second moments of a distribution. And this is not heavy tailed, but it's maybe moving toward a heavy tailed distribution, loosely speaking. The dashed line here is that quadratic Gaussian fit. You can see it does a pretty terrible job of capturing our data, but a simple mixture model of Gaussians does much better. I'm not wedded to the specific functional form of our empirical distribution, just to say that it does much, much better from a model selection, model validation point of view than the Gaussian, than the single component Gaussian. And then operationally, this is an operational definition. We're going to find the inflection point on this empirical curve that we fit for the distribution and define movements that are faster in magnitude than this inflection point as exploratory movements, and ones that are slower as exploitative movements. And another technicality is that we're going to use this term, the kurtosis, which is a measure that increases as the distribution gets larger and larger, higher order moments relative to the variance of the system. And so a higher kurtosis, 3 is a Gaussian. As the kurtosis gets higher, we'll say it's a more broad-shouldered distribution, loosely speaking. But the precise way we estimate that is with this kurtosis number. And so we can see that this is the thing that I didn't believe, that in the dark, the fish would be more Gaussian in the sense of its kurtosis being closer to 3 than in the light. And the reason I didn't think that made sense was because active sensing is driven by nonlinearities, and the fish is doing more active sensing, and nonlinearities tend to distort distributions. If you play a Gaussian distribution through a linear system, you get a Gaussian distribution. If you play a Gaussian distribution through a nonlinear system, you can get a non-Gaussian distribution. So my wrong intuition was that this ought to be more non-Gaussian than this, and the opposite was true. And so, but what we think is actually happening is that now that we understand this switching phenomenon, that when this is qualitative, when the animal is in the dark, it's doing a lot of fast exploratory movements, and only some of the time just doing pure tracking. Whereas when it's got good sensory salience, it only occasionally does these fast exploratory movements. Those are large movements, and they create the broad shoulders. And so you get these large peaks, but you still get these broad shoulders, and this becomes more non-Gaussian. And so with that idea in mind, and I mentioned this inflection point, we can use that inflection point to divide our data into fast and slow movements, and empirically calculate a state transition matrix that defines the probabilities of transitioning from, say, these, staying in the exploratory, in the exploitative mode, switching to the fast exploratory mode, or switching back, right? And again, we have no claim that this is truly a Markov process, but it's a good way of statistically representing our data. And so we have 5 fish in this study, and we measure the switching frequency for the lights being on versus the lights being off, and we see that it's switching into the fast mode much more often when the lights are off than it is when the lights are on. And the resonance time in the slow mode is much longer when the lights are on. This is true for every fish in both conditions. So each fish is statistically staying longer in the slow mode in the light and in the dark, and it's true for all 5 of our fish. And so our sort of qualitative hypothesis turns out to be true. What about another sense? We altered vision, but what if we were to go in and alter electrosense, like turn down the lights in electrosense? Well, fortunately, another lab did that for us, Malcolm McIver's lab. And what they did was, they used a set of jamming electrodes that they could turn on to decrease the signal to noise ratio. So in biology, we talk about the salience of a signal as being how strong that signal is, or the contrast. But we can think about it as the signal to noise ratio. So a low signal to noise ratio occurs when somebody plays a jamming electrode to disrupt the electroreceptive capability of the animal. And when they did that, you can think about, this is the low sensory salience, this is when it's jammed, this is the high sensory salience. You might see it sort of, this is their data, but that Debjoti has downloaded and reanalyzed using his framework. And we find something very similar, which is that when it's low sensory salience, it's more Gaussian, has a kurtosis that actually approaches 3. And when it's not jammed, the kurtosis is much higher, closer to what we saw in our just dark condition with no jamming. And so across these 2 different modalities, but still within the same organism, we see 2 trends. We see that there's this mixture of explore and exploit modes, and that the system becomes more non-Gaussian, more infrequently exploratory when the signal noise ratio is higher. Okay, so is that true just for eigenmania? After all, they want us to find their eigenvalues, so maybe that's just true for them, or is it true for other organisms? unbelievably, Deb Jyoti went through an incredible amount of literature, downloading papers, downloading data, emailing authors, and analyzing their data. And what I'm going to show you, all of the data that he's downloaded and analyzed to a data set that he was able to find tasks like this. Every single one supports our hypothesis. I'm not cherry picking the few that worked. It's an amazing set of observations that he's made. And he's done this for animals from bacteria to amoeba, even plants, C. elegans, Drosophila, several of which with the asterisks have multiple different levels of sensory salience, which is really important for our hypothesis, and over a huge range of sensory environments: phototaxis, chemosensing, temperature sensing, molecular sensing, olfaction, auditory, touch, and vision, as indicated. And these are just the common names of the animals that he's studying. And so I'll just give a couple of quick examples, and then I'll be done. So one example is medial lateral postural sway, where somebody stands balancing. They have their finger on a device that gives them a sense of touch feedback, but they're not holding on, it's just for touch feedback. And they have a visual scene in front of them to help them balance. They do this sort of feet aligned in order to decrease the mechanical stability and exacerbate the postural sway. I should note that actually very recently a paper came out showing that movement of the proprioceptors is important for their ability to measure your postural state. So active sensing has been shown to be needed for these proprioceptors, okay? So, well, what he found was, once again, the postural sway matches this hypothesis. So the humans tend to be doing this postural sway. I don't think I'm going to show this data, but in addition, when you take away the touch, it becomes more Gaussian. So when you decrease the overall signal noise ratio by moving one of the sensory modalities, you see a diminishing of this kurtosis value toward 3 and a broadening of the overall distribution, a decreasing of the relative peak of the central peak. Another really incredible system is this hawk moth proboscis task, where the hawk moth is feeding from an artificial flower. I was actually doing a sabbatical in Tom Daniel's lab when he was talking about how he was 3D modeling in Mathematica the shapes of these flowers and printing them in order to look at proboscis tracking. I had no idea that my postdoc would be analyzing that data someday, but this is an example trajectory of the proboscis tip as it's trying to enter into the flower nectary. And inside the bottom, there's a sugar liquid reward. Okay, that's what the moth is going for. And once again, we see this beautiful mixture model emerge from the velocity of the antenna tip in degrees per second on the x-axis there, and probability on the y-axis. And another cool finding is that the flatter the printed flower, which is less salient local curvature, the more Gaussian it becomes, or the more Gaussian it becomes, because it's a less salient signal. So, so that's just 2 of the organisms that we looked at. We looked at many more. This is just a sampling of them. They all show this mixture model distribution. And you can see that quite clearly from these examples. And so one more example showing sensory salience. This is a cockroach looking for a sex pheromone. It's coming up when, and you see this mixture model for the normal condition. But what then these authors did was they flipped the antenna and did the same experiments. And we see that the animal becomes closer and closer to Gaussian as it loses its antenna. I mean, it has a very low signal noise ratio. Now it's just casting back and forth, doing mostly active sensing, and the task-oriented movements are much less. So, so this all suggests that this broad shoulder distribution and its dependence on sensory information is really driven by, is found independently throughout evolution, because it's not likely that we develop the same mechanisms as an amoeba or a bacterium. It's really more likely to have been found independently. And so I'll just say that in closing, that we've modeled this behavior using a very simple state estimation-based controller where we inject an active sensing signal. And we've tried both the persistent excitation and a switch strategy that Andy Lampersky came up with, which is very simple, which is that when the trace of the covariance matrix of your extended Kalman filter reaches an upper threshold, you turn on active sensing, and when it reaches a lower threshold, you turn off active sensing. And that both reproduces the distribution as well as does better from an energetic point of view than this persistent excitation strategy. And it recovers, importantly, this idea that as we decrease the signal noise ratio, our kurtosis in that algorithm goes to 3. It becomes a Gaussian distribution. And so hopefully that was all clear. I'll just skip through the summary here in the interest of time, and just say that a number of people contributed to this. I didn't have time to find the updated, go back and find the right historical timeframe photo to capture my trainees from here, but he was instrumental in helping, Balav was instrumental in helping with the videography. Many people provided us data, and these are the co-authors on the study. It's available online and currently in review. And this is this co-author of our study on the nonlinear observability properties. And with that, I'd be happy to take any questions. And if you don't have time to talk to me today and are curious, please feel free to reach out.